Usually when we talk about a medical diagnosis, there is this expectation of precision, like engineering almost.
Right, yeah.
You break your arm, the X-ray shows that jagged white line, and the doctor just points at it and says, there it is.
It's binary. I mean, it's broken or not broken. It's a very clean narrative.
Exactly. But then you step into the world of cardiac repair, and suddenly that X-ray machine is kind of useless. We are looking at a healing landscape that is, well, honestly, it's murky.
Extremely murky.
So today, we are attempting to clear that murkiness up for you. We are diving directly into a highly detailed 2026 paper from Cell Reports Medicine. It's by Stjonglis and colleagues. And our mission here, we are exploring a fundamentally different approach to treating myocardial infarctions, which, we commonly call heart attacks.
And we should clarify, we aren't talking about stents today.
Right, no stents. We aren't talking about bypass surgeries either. We are looking at using trace metals, specifically zinc and copper, and using them as a highly sequenced rescue crew for the heart.
Which is fascinating. Just to give that some overarching context for you listening, trace elements make up less than 0.01% of our body weight.
Wow. That's practically nothing.
It's a microscopic fraction.
Right.
But you can think of them as the spark plugs of our cellular engines. I mean, they trigger gene expression, they facilitate protein synthesis, they literally keep the metabolic engine running.
So they punch way above their weight.
Oh, absolutely. And what we are unpacking today is how mapping the exact biological timing of these trace elements post-heart attack has led to, well, a revolutionary new treatment. Because timing, as we will see, is quite literally the difference between life and death for heart tissue.
Okay, let's unpack this with a mental image. Think of the heart after a massive heart attack as a natural disaster zone.
I like that analogy.
Right. So a hurricane just swept through a city. To rebuild, you need specific materials. But if the drywall and the lumber arrive while the floodwaters are still raging and before the foundation is even poured, it's completely useless.
That creates more debris.
Exactly. So what exactly happens to these trace elements in the heart the moment it is deprived of oxygen?
Well, the biological timeline here is terrifyingly fast. I mean, the moment a heart attack occurs, the blood flow stops, and oxygen is instantly cut off.
Right.
Within the first 24 hours, the infarcted area, which is the dying tissue, is flooded with severe inflammation and reactive oxygen species, or ROS.
Okay, wait. I always hear about reactive oxygen species in the context of, like, anti-aging skin creams. But in a heart attack, they're much more violent, right?
Far more violent. You need to think of reactive oxygen species as microscopic shrapnel just bouncing around inside the cell.
Oh, wow.
Yeah, they are highly unstable molecules that are missing an electron. So they violently rip electrons off of anything nearby. Cell membranes, DNA, proteins.
So they're just tearing the house apart looking for parts.
Exactly. It causes massive oxidative stress, literally shredding the cellular infrastructure.
And this is where zinc enters the picture to stop the shrapnel.
Zinc doesn't just enter the picture. It becomes the single most critical asset in the microenvironment. Zinc acts as a vital cofactor for your body's natural antioxidant enzymes.
Okay.
We're talking about heavy-hitting enzymes like glutathione peroxidase and superoxide dismutase.
Those are some big words. Basically, they are essentially the magnetic nets catching all that cellular shrapnel, right?
That is an excellent way to picture it. But because these enzymes are working absolute overtime to fight the oxidative fire, the free zinc levels in your system plummet drastically.
Because it's all being used up.
Right. The researchers mapped this and found that zinc drops to its absolute lowest point within 24 hours. And it stays dangerously low for 4 to 5 days. It is completely consumed by the battle.
So the zinc is depleted, putting out the fire. But if we go back to our natural disaster analogy, eventually the floodwaters recede. The blueprint has to change from, emergency response to actually rebuilding.
And it does. Around days 5 to 7, the biological priority shifts. The heart desperately needs new blood vessels to bypass the dead tissue and survive.
Which is angiogenesis, right?
Correct, angiogenesis. And to trigger this, the body's copper levels suddenly spike.
Okay, so where is all this copper going?
The body mobilizes massive quantities of copper into the endothelial cells, which are the cells that line your blood vessels. This copper activates a master protein called HIF1, or hypoxia-inducible factor 1. HIF1 is essentially the cellular foreman that yells, "Hey, start building new pipes." And to pull this off, specific copper transporters work overtime to funnel copper straight into the oxygen-starved site.
But wait, heavy metals floating around the heart sounds kind of dangerous. If we connect this to the bigger picture of homeostasis, there has to be a catch. What happens if the copper arrives too early while the inflammation fire is still raging?
It is disastrous. If copper stays too high for too long, or if it spikes during that initial 5-day inflammatory phase, it acts as an accelerant.
Like throwing gasoline on the fire?
Exactly. It triggers massive oxidative stress and induces apoptosis, which is programmed cell death. The copper literally commands the remaining struggling heart cells to self-destruct.
So let me do a quick recap here for you listening to make sure I'm tracking this biological roller coaster right. For the first 5 days post-heart attack, your zinc plummets because it's desperately being used to fight the oxidative fire.
Right.
Then, right around days 5 to 7, copper spikes to act as the foreman laying down new blood vessels. Imagine being the listener right now, realizing that the basic trace minerals sitting in your kitchen cabinet are currently operating inside you as this highly coordinated, sequenced rescue crew. I mean, it completely reframes how you look at the human body.
They really aren't static building blocks. They're dynamic, highly timed responders.
Which perfectly sets up the massive failure of past therapies. Because, scientists have known for a while that zinc and copper are involved in heart attacks. But here is where it gets really interesting. Past researchers tried to fix the damage by just injecting patients with a blended cocktail of metals, just dumping both zinc and copper into the bloodstream at the exact same time.
And the therapeutic efficacy of those cocktails was wildly inconsistent. It mostly resulted in failure. And based on what we just discussed about the timeline, you can probably see why.
Well, yeah. I mean, if you inject a cocktail of zinc and copper on day 1, you are sending plumbing materials into the middle of a burning building. You are delivering copper during the peak inflammatory phase, which you just said triggers the cells to self-destruct.
That's right. You are pouring the roof before the foundation is even dug. It ignores the heart's strict biological schedule entirely.
So how did Zhang and the team in this 2026 paper actually solve this? I mean, how do you control the timing inside the body?
They introduced a radically intuitive concept called sequential therapy. They designed a regimen that meticulously mimics the heart's natural pathological timeline.
Okay, so matching nature.
Exactly. They provided zinc, and only zinc, from day 1 to day 6. Then, they completely cut off the zinc supply and switched exclusively to copper from day 7 to day 14.
And they didn't just use standard liquid IV drips either, did they? They used something called silicate bioceramics.
Specifically, a mineral called hardystonite to deliver the zinc, and cuprorivate to deliver the copper.
Hardystonite and cuprorivate, okay.
Right. And they chose these specific bioceramics because as they slowly break down the body, they release silicon ions alongside the zinc and copper.
Wait, what's the role of the silicon? Why add another element?
Silicon acts as a biological enhancer or an amplifier. It synergizes with the metals. It basically pries open the cellular doors and significantly improves the cell's ability to uptake and use the zinc and copper.
Oh, that is clever. And the results in the rat models were absolutely undeniable. They measured a whole suite of cardiac functions. To give you some context, they measured LVEF, left ventricular ejection fraction, which is essentially a percentage of how much blood the left ventricle pumps out with each squeeze.
All right, a key metric for heart health.
And they measured LVFS, fractional shortening, which looks at the actual inward movement of the heart walls. Both of these mechanical squeezing metrics improved massively with the sequence.
They also measured the physical deformation of the heart chamber, because after a severe infarction, the dead tissue turns into a very weak scar.
Like a thin spot on a tire.
Exactly like that. And under the constant pressure of pumping blood, that weak scar wall bulges outward and stretches. The sequential therapy significantly reduced this bulging. It preserved the physical architecture of the heart.
But my absolute favorite part of this study, like the true aha moment for me, is what happened when they messed with the sequence just to prove their point.
The control group.
Right. They created a control group where they reversed the order. They gave copper first from days 1 to 6, and then zinc from days 7 to 14. I mean, we know copper early acts as an accelerant to the fire, so let me guess. It tore the surviving cells apart.
Total failure. The therapeutic benefits vanished completely. The heart function at day 28 was statistically no better than if they had received zero treatment at all.
Wow.
It proves that biological timing isn't just a clinical suggestion. It is a strict biological law.
Okay, so to help you keep this timeline straight, I've got a quick mental mnemonic for you. It's ZACB. Zinc for antioxidants early, copper for blood vessels later.
I like that, ZACB.
So just to pause and summarize our second major point here. Previous cocktail therapies failed because they dumped metals into the system all at once, totally ignoring the schedule. The proven sequence is zinc for 6 days, followed by copper for week 2. And if you reverse that order, you actively ruin the heart.
It really forces you to question the broader cultural obsession with the all-at-once approach to health.
Oh, totally.
Like taking massive blended multivitamins, regardless of what your body actually needs in that specific moment.
It really does. But let's dig into the how. We know the sequence works, zinc, then copper. But how is the heart tissue actually translating these raw metal ions into a healing response? Because I read the methodology here, and the master switch they found is a gene I have only ever associated with jet lag.
Ah, yes. You are referring to PER2, period, circadian protein homolog 2. This discovery is perhaps the most groundbreaking finding of the entire paper.
A circadian rhythm gene inside the heart muscle.
Yeah, we usually think of circadian rhythms as just sleep-wake cycles controlled by the brain. But individual organs, the liver, the kidneys, the heart, they have their own local internal clocks.
Wait, really?
Oh, yeah. The heart's metabolic needs change drastically depending on whether you are sleeping or running a marathon, so it needs a local timekeeper.
So what happens to the heart's local clock during a massive trauma, like a heart attack?
The trauma essentially shatters the clock mechanism. The researchers found that immediately after an infarction, PER2 levels in the heart absolutely plummet. The local circadian rhythm is broken.
And how does dropping a bunch of zinc and copper onto the heart fix a broken clock?
The metals act through the immune system. During the early inflammatory phase, the heart is swarming with M1 macrophages. Think of M1s as the cellular demolition crew. They clear out the dead tissue.
Okay, M1 is the demolition crew.
Right. And the zinc specifically targets these M1 macrophages and stabilizes their PER2 expression. Later, during the repair phase, the landscape shifts to M2 macrophages, which are the reconstruction crew.
And so the copper targets the M2s.
Exactly. The copper then targets those M2 cells, boosting their PER2.
Okay, so the metals use the immune demolition and reconstruction crews to reboot the heart's clock. But why does the clock matter so much for survival? Like, it is great that the heart knows what time it is, but how does that save a dying cell?
Because PER2 isn't just a timekeeper. It is a massive metabolic switch. Normally, a healthy heart gets most of its energy by burning fat, a process called fatty acid oxidation. But burning fat requires a tremendous amount of oxygen.
Which an infarcted, blocked heart simply doesn't have.
Exactly. An oxygen-starved heart attempting to burn sugar will stall out and die. What PER2 does is force an emergency metabolic shift. It reprograms the mitochondria to stop burning fat and start burning glucose sugar instead.
Why sugar?
Because glycolysis, which is the burning of sugar, requires much less oxygen. It provides an immediate life-saving energy lifeline to the suffocating cells.
That is incredible. The clock gene is literally an emergency fuel selector switch. But mechanically, how does PER2 force the cell to stop burning fat?
It operates by suppressing a specific cellular pathway called PPR gamma. PPR gamma is basically the master switch for fat storage and fat metabolism. By aggressively suppressing PPR gamma while simultaneously boosting HIF-1 alpha, PER2 corners the cell.
It corners it?
Right. It shuts down the fat-burning machinery, leaving the cell with absolutely no choice but to consume glucose to survive.
And the researchers proved this definitively right. I saw they used an AAV9 virus vector. So they essentially used a virus as a microscopic Trojan horse to go in and manually edit the genes.
Precisely. First, they used the viral Trojan horse to knock down or basically silence the PER2 gene in the heart. When they deleted that clock gene, the sequential zinc-copper therapy completely stopped working.
Even with the perfect timing.
Even with the perfect timing, the heart failed. But then they used the virus to artificially overexpress the PER2 gene. They forced the clock to turn on. And when they did that, the heart shifted its metabolism and healed itself significantly, even without the trace metals.
Wow. So the metal ions are just the keys. PER2 is the actual engine of survival.
Exactly.
So let me wrap my head around this for a quick summary. Your heart has its own clock, driven by the PER2 gene, which breaks during a heart attack. Zinc and copper sequentially step in to stabilize this clock through different immune macrophages. And by stabilizing the clock, it suppresses the fat-burning pathways, forcing the oxygen-starved tissue to burn sugar just to survive.
You nailed it.
Knowing that your heart literally changes its fuel source under stress really makes you view your own metabolism differently, doesn't it? It is not just calories in, calories out. It is a desperate calculation of what fuel costs the least amount of oxygen.
It elegantly highlights how deeply intertwined time, immunity and energy metabolism truly are.
Okay, so we have the sequence, which is zinc, then copper. We have the mechanism, the PER2 clock shifting the fuel source. But I have a major pushback here.
Go for it.
In the early experiments we just talked about, they were injecting these metal ions into the tail vein of rats. Systemic delivery. That feels like crop dusting an entire farm just to water a single dying plant. The metals go everywhere. They get flushed out by the kidneys super fast. How on earth do we get these trace elements only to the damaged part of the heart in a human?
And that is the ultimate clinical hurdle. Systemic tail vein injections result in massive physiological waste. And as we noted, high systemic copper can cause off-target toxicity in the liver or lungs.
Right, you don't want heavy metals building up in your liver.
No, you definitely don't. So to solve this, the researchers engineered a delivery system called the PerMed device.
PerMed, what does that stand for?
It stands for Perfusible Multifunctional Epicardial Device. Visually, you can imagine a highly engineered, flexible 3D printed patch.
And a surgeon physically attaches this to the heart.
Yes, the patch is sutured directly onto the epicardium, the outer surface of the heart, placed exactly over the dead infarcted tissue.
So it is a physical patch. Does it just hold the medicine or what?
It serves two separate vital purposes. First, remember earlier how we discussed the heart wall bulging and dilating because the scar tissue is weak?
Yeah, the thin spot on the tire.
Right. The PerMed patch provides immediate mechanical support. It acts like an external structural girdle, physically holding the weakened left ventricle together to prevent it from stretching out.
Oh, that makes perfect sense. Structural scaffolding. But what about delivering the sequenced metals?
That is the second function. The patch is connected via a microscopic tube to an implantable osmotic pump. This pump automatically pushes the exact sequence, 6 days of zinc, followed by copper, directly into the patch.
Oh, wow.
And then the patch slowly diffuses the ions in situ, right on site. It creates a highly concentrated, localized ion pool directly over the damaged tissue without spilling into the general bloodstream.
And because you aren't crop dusting the whole body, you can use way less of the metal, right?
Dramatically less.
Huh.
They only needed to use 25%, 1/4 of the dose compared to the systemic injections.
That's a huge reduction.
It is, yet because it was delivered directly into the microenvironment, it achieved much higher, vastly more stable local concentrations. The kidneys just couldn't flush it away.
That is brilliant engineering. And the imaging they used to prove this was intense, too. They used a PET-CT scan utilizing a 17-segment heart model, right?
Yes. Cardiologists divide the heart into a 17-segment map to pinpoint exactly which zones of the muscle are dead, struggling, or healthy. The PET-CT imaging allows you to look at the regional sugar metabolism of the heart in 3 dimensions.
And it lit up.
Unprecedented results. The localized PerMed delivery caused a massive surge in local cardiomyocyte metabolic capacity right under the patch. But even more impressive, they looked for specific proteins, CD31 and alpha SMA.
And what do those do?
These proteins act as the literal cellular scaffolding for new blood vessels.
So seeing those proteins meant the pipes were successfully being laid.
Exactly. The patch was driving robust new blood networks deep into the dead zone.
I also saw they measured the actual makeup of the scar tissue itself, the collagen ratios.
Yes, the ratio type 3 to type 1 collagen. Think of type 3 collagen like a stretchy rubber band. It is elastic and highly favorable for early tissue repair.
Okay, rubber band is good.
Type 1 collagen, however, is more like stiff concrete. Too much of it leads to rigid fibrosis that stops the heart from beating properly. The localized PerMed therapy significantly improved this ratio, meaning the heart formed a healthier, highly flexible scar.
So to bring this all together for our final summary, systemic injections waste materials and risk poisoning other organs. But this PerMed patch creates a localized micropharmacy. It physically holds the heart together while perfectly timing the drug delivery, requiring only 1/4 of the dose. I mean, imagine the implications for this.
It's massive.
Having a timed micropharmacy attached directly to an organ could completely change how we treat highly localized diseases, like targeting solid tumors without destroying the rest of the body with heavy treatments.
It really represents the pinnacle of precision medicine, delivering the right element at the exact right biological hour to the exact millimeter of damaged space.
So what does this all mean? Let's zoom out. We started by mapping a natural disaster timeline inside the heart. We saw zinc drop to fight the oxidative shrapnel and copper spike days later to lay the new pipes. We learned that to save the heart, we have to respect that sequence. Zinc for the first 6 days, copper for the next week.
And we uncovered the master switch, the PER2 circadian clock gene, which uses those sequenced metals to forcibly shift the heart's metabolism, shutting down fat burning to save oxygen, and relying on sugar burning to survive.
And finally, we saw how the PerMed patch delivers this exact sequence locally, preventing the whole systemic crop dusting effect.
This body of research forces us to completely rethink the fundamental definition of medicine.
How so?
Well, for centuries, medicine has been focused almost entirely on what drug you take. What is the active compound? But this proves that what is only a fraction of the equation. It is equally about when and where exactly it is deployed in the body's microenvironment. A cure given on Tuesday can literally become a poison if given on Thursday.
A cure on Tuesday is a poison on Thursday. That is wild. Which brings me back to our opening thought. We wanted the comfort of a clean X-ray, the binary of broken or fixed. But biology isn't a static snapshot. It's a moving picture. It is a ticking clock.
Exactly right.
So I want to leave you with one final mind-bending thought to mull over. If our individual organs have such precise, localized circadian rhythms clocks that dictate exactly which trace elements they need down to the exact day and hour just to survive, what happens when we blindly swallow a blended multivitamin pill every single morning?
It's a great question.
Are we accidentally bombarding our body's delicate, highly choreographed spatiotemporal balance with ions at the exact wrong time of day? Thank you so much for joining us on this deep dive. Until next time, keep questioning the timing of everything.